Değerli katılımcılar, sevgili arkadaşlar, ülkemizin bilim dostu, bilgisever, güzel insanları. TÜBİTAK Temel Bilimler Araştırma Enstitüsü'ne, seminerimize hoş geldiniz. İnsanoğlunun canlı alemin özünü anlama çabaları yüzyıllar boyunca devam etmektedir. Anlamak her zaman güzel bir nimettir. İnsanlık için yeni fırsatlar, mutlu yaşam, güzel gelecek demektir. Bugün gelinen noktada bir hücrenin tüm canlıların en küçük yapı taşı olduğu iyi bilinmektedir. Canlıların tüm yapısal ve işlevsel özelliklerini belirleyen hücrelerdeki moleküler mekanizmalardır. 1960. yılların sonlarına doğru ABD uzay programında inanılmaz başarılar elde etmiş ve insanoğlunun Ay'a ayak basmasını sağlamıştır. Çünkü Ay'a gidecek kadar ve fazlasıyla temel bilgi üretilmiş ve buna dayanarak ortaya konulan mühendislik imtihanı başarıyla gerçekleştirilmiştir. Ülkesinin bu başarılarından yola çıkan Başkan Richard Nixon, çağın hastalığı kanseri yenme programı başlatmış. Fakat bu program başarıya ulaşamamıştır. Bunun nedeni yaşamın moleküler temelleriyle ilgili yeterince temel bilginin üretilmemiş olmasıydı. Temel bilimin yaşamın moleküler düzeyde işleyişine ışık tutan devrimsel bilgi üretimi günümüzde hızla devam etmektedir. Örneğin biyolojik bilimlerde ve teknolojideki gelişmeler bizi hücrelerin ve dokunun fenotipinin mühendisliğiyle hastalıkların tedavi edilebileceği bir çağa yaklaştırmaktadır. Bu gelişmeler hücrelerin ve dokuların işlevini manipüle etmeyi, genleri, hücreleri ve çevrelerini etkileyen stratejiler kullanarak öngörülebilir bir şekilde davranmalarını mümkün kılmaktadır. TÜBİTAK Temel Bilimler Araştırma Enstitüsü, temel bilimin diğer alanlarında olduğu gibi biyolojik bilimler alanında da en üst düzey ve yenilikçi bilgileri ülkemizin bilim ve eğitim camiasına aktarma yönünde yoğun çaba göstermektedir. Biyolojik bilimler seminer serimiz bu amaçla düzenlemektedir ve bu akşam Harvard Üniversitesi'nden dünyaca ünlü bir bilim insanı Profesör Shamil Sunyaev'in katılımıyla bir güzel bölüm daha gerçekleştirmek üzere bir araya gelmiş bulunmaktayız. Profesör Shamil Sunyaev, İnsandaki Mutasyonun İstatistikleri ve Biyolojisi başlıklı bir konuşma yapacaktır. Ben de bu vesileyle hepinizi saygı ve sevgiyle selamlıyorum. Seminerimiz İngilizce olarak gerçekleştirilecektir. Dear Professor Shamil Sunyaev, dear participants, dear colleagues and friends, a very warm hello everyone and welcome. Welcome to the online Biological Sciences Seminar Series of TÜBİTAK Research Institute for Fundamental Sciences, which we organize for national and international audiences with the participation of distinguished scientists and great speakers of the world. We organize these seminars relying on the unifying feature of science, especially in this difficult period of life when the coronavirus outbreak continues to evolve in the world. It is my sincere pleasure to emphasize that this evening we are absolutely thrilled to have Professor Shamil Sunyaev with us, a distinguished scientist and a very special speaker from Harvard University. He has kindly agreed to join us for this seminar, and he is going to give a great talk, Statistics and Biology of Human Mutation. At the end of the talk, we will have Q&A session. Please ask questions by sending a message using the chat button or just by raising hand. Shamil Sunyaev is a professor and distinguished chair of computational genomics at Harvard Medical School and the research geneticist at Brigham Women's Hospital. He is also an associate member at the Broad Institute of MIT and Harvard. Research in his lab encompasses many aspects of population genetic variations, including the origin of mutations, the effect of allelic variants on molecular functions, population and evolutionary genetics, and genetics of human complex and Mendelian traits. He developed several computational and statistical methods widely adopted by the community. Shamil Sunyaev obtained a PhD in molecular biophysics from the Moscow Institute of Physics and Technology and completed his postdoctoral training in bioinformatics at the European Molecular Biology Laboratory. He received the International HDL Research Award by Pfizer and the John and Virginia Taplin Award from the Harvard MIT Division of Health Sciences and Technology. Professor Sunyaev awarded funding from National Human Genome Research Institutes. He co-leads Genome Sequencing Program Analysis Center and is actively involved in the Undiagnosed Disease Network and in the Brigham Genomic Medicine Program. He also co-organizes the Boston Evolutionary Genomics Group. With this, I want to thank once again Professor Shamil Sunyaev for joining us and invite him to the stage to begin his talk. Shamil?
Yes, I'm sharing my slides. Can people see the screen?
Yeah, it's okay.
So, Alikram, thank you very much for the introduction. So the talk is going to be about mutations. So you mentioned the pandemic. I'm not going to talk about viral evolution. We have some projects now on COVID. So this is primarily about mutations in humans. And it's great pleasure to be here. It's a pity we're doing this in the virtual format. So hopefully it would be possible to visit and present a normal physical talk in person. Okay, so the current story is about statistics and biology of human mutation, and it is what it is. I'll talk about statistics first, and after that about biology we learned. It is my understanding that this is a research seminar. It's not really popular talk, so this is about our recent research work, and I will try to emphasize the computational aspects and also aspects of biology. So the whole story is about de novo germline mutations in humans, and this means differences between parents and children. As we know, we primarily inherit DNA from our moms and dads, and this is almost true, except there are some mutations that happen in DNA, and I'll be talking specifically about point substitution. So when one letter of the DNA alphabet changes in a certain position in the genome. And these events are almost unbelievably rare. What we know about the rate of mutation is it happens around 10 to the -8 per nucleotide per human generation, right? So it is very rare that these events happen. However, they happen sufficiently frequent that every individual human would harbor about 70 of these events, right? So most genetic differences between individuals are inherited differences. They are not de novo, but some are. And the major question is how these events happen, both why they happen so rarely and also what are the mechanisms which govern occurrence of these mutations. This is the distribution, and this comes. This plot comes from Icelandic data from family sequencing. It was developed a few years ago. And what you see here, it's, as I mentioned, around 70 is the average number. And the distribution is a textbook Poisson. So there is not a lot of variation in the population, except there are dependencies on ages of father and mother. So all the parents, children of older parents have slightly more mutations than children of younger parents. But generally, it's a nice Poisson-looking distribution, and the average is several dozens of mutations per individual. So this is what all of us have as differences between our genomes and genomes of our parents. And some basic facts about human biology here. So of course, one human generation involves many cell divisions. So these are cell divisions along the developmental trajectory. In the trajectory towards germ cells. These are some cell divisions of germ cells. And what's important here is that there is substantial differences between the two sexes because male germ cells continuously divide through life. However, oocytes, female germ cells, are arrested in phase 2 of meiosis and do not divide. And that's a key observation because even epidemiologically, I'll show some molecular evidence of this, but this suggests that some of the mutations do not arise as a product of cell division. They arise in the non-dividing arrested cells. And possibly as a consequence of that, there are more mutations that arise in fathers than in mothers. And I'm talking only about point mutations, changes of individual nucleotides. Why study mutations? There is a lot of interest in mutations. And the primary reason is that accurate transmission of genetic information is the fundamental properties of life. As you see here, DNA goes through replication, it's being divided, and so cells are dividing and DNA is copied. So there are 2 copies of the original molecule, and the precision of the process is amazing. The number of errors is at most 10 to the -10. However, because there are some errors, these errors allow us to learn about maintenance of the genome, about processes of DNA replication, DNA repair, how does the cell maintains genomic integrity, how lesions are repaired, and how DNA replication process is ongoing. So one reason to study mutations is pure basic biology. This is not the only reason. Obviously, mutations generate genetic diversity. And my lab is very interested in genetic diversity. And this example, of course, is not human. We have almost any population of almost any species is genetically diverse. So we have varieties. And these differences lead to differences in phenotypes, right? In why some people have higher blood pressure than others and different values of different traits. So mutation is a key player because without de novo mutations, genetic diversity wouldn't exist. Mutations fuel evolution, right? So no evolution is possible without mutations. And I know having Darwin finches on the slide is almost a cliche. But what happens here is that evolution is a change leading to eventual speciation. And this change is fueled by mutational process, again, because evolution is driven by selection on genetic variation. There is no variation in the absence of mutation. And also mutations cause genetic disease. And I mentioned complex phenotypes that are primarily determined by variation segregating in the populations, like common diseases. However, many rare Mendelian diseases are caused directly by de novo events. So out of the 70, if a mutation lands on a very unfortunate position in an important gene, you may have an immediate presentation of a disease phenotype. We and others are studying these mutations and trying to use genomics to understand load of genetic disease and diagnose these patients. This is not what I'm going to talk about, but As a motivational slide, there are multiple reasons to think about mutations, and this ranges again from understanding diversity, from evolution, and from disease itself. So how do we go about studying mutations? And this is an interesting from purely methodological perspective, because historically, mutagenesis was studied in experimental systems, and frequently this would be a reporter assay, this would be some system amenable to manipulation, for example, yeast. And you can generate some mutagenic process, and you can analyze specific mechanisms that create mutations. However, this would be limited to one locus, and this would be limited to one system and one organism in certain and specific conditions. So it's absolutely impossible to say whether these mutagenic events are actually relevant to what's happening in humans. The alternative was offered by the genomic analysis, shown here on the left. And in genomic analysis, this started with comparative genomics, with comparing human genomes to the human genome to genome of primate species, where we can see sequence changes, and looking at statistical properties, we can think about properties of mutational data. And we know that these mutations actually happen. So this is something that occurs in nature, in this case, in the human lineage. However, we have no idea about mechanisms, right? So looking at statistical patterns by themselves is not very informative about mechanistic biology. Again, experimental work in manipulated systems discovers mechanisms of mutagenesis, but by itself is unable to establish their relevance. And now we have plenty of data sets. We have computational analysis of species divergence, as I just mentioned, population variation, de novo germline mutations, and this is the topic of my talk, somatic mutations. So not mutations happening between generations, but mutations happening in healthy cell, or mutations happening in cancer cells. So we have plenty of data. However, when we characterize patterns of mutations, we cannot propose any mechanisms. So that's a methodological complexity. Now I have to tell you that in the beginning of our entry into the field, I was pretty naive. I thought that I would go to a meeting with people studying mutations and actually understanding biochemistry, and I would show statistical patterns, and these people would tell me, oh, I know this corresponds to this specific biology, and we're done. Now, the truth of the matter is that you show at such a meeting, and there are several hundred people, and all these people study mutational mechanism, and they have their own systems, and they have their favorite mechanism, and you show them statistical patterns, and they tell you, oh, this is my mechanism, I think this is what's happening. So again, it is almost impossible just to naively match statistical mechanisms to specific biological, statistical patterns and statistical observations onto biological mechanisms. And this goes all the way up, right? So even the very basic question, how many mutations occur as nucleotide misincorporations during DNA replication versus mutations initiated by DNA damage that is either being bypassed during replication or being misrepaired. This question is open, so we don't really know what is the partition of human mutations into these two broad categories, right? So some people, I think, assume that all mutations happen as the result of replication infidelity. However, we don't believe that's the case, but it is very difficult to quantify. There was some progress, and the progress was offered by cancer genomics. In cancer genomics, so again, what people analyze in cancer genomics, these are primarily somatic mutations. So mutations that are not in germ cells, and not those mutations that are transmitted between generations, but mutations originating in one cell and transmitted to the next cell generation. And eventually, if cancer spreads, this cancer becomes clonal. So there is such a thing as cancer genome. All of the cells share the same genomic information, and they share mutations happening early in the progenitor of this tumor. So the idea that was proposed and that got traction in the field is that you have specific nucleotide changes, and you can think about the context. So we know that frequency of mutations of specific, so letter C mutates to letter A, this frequency of this process depends on nucleotides that are left of this letter or right of this letter, or as we say, 5' and 3' direction. So adjacent, immediately adjacent neighbors. And we can represent the all mutations in cancer using this information, and we can analyze it with respect to differential exposure, right? So we have, for example, melanoma that is caused by UV light. We have lung cancer caused by smoking and so forth. And we can find specific signatures, so specific vectors in this 96-dimensional vectors corresponding to all these contexts, corresponding to these exposures. So in this case, to UV or to smoking. So the way this is done is that we model that data in the cancer genome, not we, this is of course work of others, is composition of different processes that have distinct spectra, and the resulting data is superposition of these processes. So we have differential exposure is just statistical instrument, right? So we're just analyzing different patients' genomes, and this analysis can be formalized as multiplication of very large matrices. So we have metrics corresponding to all the processes by their spectra. We have a matrix corresponding to intensities of processes per individual, and a product of these matrices would give us all the data we observe, meaning all trinucleotide mutations in every single patient. And the process in the inference is provided by factorization of this matrix product. Now, I'm not going to talk about complexities of factorization of matrix product. In our work, this specific method didn't work, so we had to rely on a newer machinery. But this was the basic idea in cancer genomics, and this provided a lot of insight connecting epidemiological exposures to mutational process in cancer. However, this specific analysis is not applicable to germline genetics because all individuals are mostly exposed to the same set of exposures, and spectrum between individuals are not very different. So this doesn't really help us to understand biology of germline mutations, so differences between parent and children rather than differences between tumors. So as I said, this is a research talk, so I'll be talking about a recent paper just out in Science, and this will be about application of such a method to germline data. So what we decided to do, we decided to come up with the following model, and working with large data sets, and this is what I'd like to explain very clearly. You can work with very basic set of theoretical assumptions. So the assumption we're making is that mutation rate is variable along the genome, we know that. However, we think that at every genomic position, mutations we observe are produced by a number of processes, and we're seeing some of this, of the effect of these processes, right? So if we look at different intensities of the processes at every locus and differences in the spectra, we can decompose mutational data and learn about process variation along the genome. So this means that we offer a spatial model. We have a genomic coordinate here, so this goes along the chromosome, and we assume that various processes are variable in different scale. Now, again, I make no assumptions about specific properties of this variation, right? So I don't know the scale of this variation. The only requirement is that the scale is not too short, otherwise I have no data. I'm not making any assumptions about degree of this variation, and I'm not making any assumptions about the number of those processes, right? So the only assumption is that these processes are relatively independent, so I can model my mutational data as additive over processes. And I know that these processes are variable along the genome. So this is the cartoon about the methodology. So imagine that I have, these are single nucleotide mutations. Of course, we work with full trinucleotide alphabet. But imagine that we have this process one, and this is a hypothetical process that generates few mutations A to G and many mutations C to T. And I have another process that generates more A to G and less C to T. And this process has variable intensity. Process one, this hypothetical process one has variable intensity along the chromosome, and process two has also variable intensity, and they are not the same. So when I look at mutation rate dependency per position, I would see that this is the intensity generated by, these are mutations, C to T and A to G, generated by process two. And these are mutations C to T and also A to G generated by process one, right? So this is what I observe in the data as generated by process two, and this is what I would observe in the data generated by process one. And in total, I would observe all the mutations generated by both processes, and they would be also variable along the genome. So I can cut this up into genomic windows and represent intensity of each process at each window as a raw vector, right? So I just write down what is the intensity of the process in a genomic window. And I can have column vectors that would represent intensity of the specific mutation produced by each process. So it would be different types of mutations, and each mutations will have differential intensity, but only those produced by a given process. So then my data will be given as a product of these two matrices. And I will have to decompose this matrix product. I would have to factorize this matrix product to identify both spectra and intensity. Now, this is the crucial point. In this product, I have none of these values are known, right? I have no idea about spectra. I have no idea which processes generate which mutations. I do not. And I have no idea about intensities of processes along the genome. So I don't know any of this. The only thing I know is the data. So this means that given that I have sufficient amount of data, I can learn both characteristics of mutational processes and their variation along the genome. And I'm not going to talk about specific methodology and uniqueness of solution and other issues because this is probably too technical for this presentation. Another technical idea here helps us to determine whether processes are correct, and also help us to determine how many processes we need. And this is the idea about asymmetry between DNA strands. So we know that DNA strands are symmetric. However, various biological events, such as such as transcription, breaks the symmetry, right? So in transcription, I have a coding strand and non-coding strand, and they are different from each other, right? Just because of direction of our RNA polymerase. And in the replication fork, I also have a leading strand and lagging strand during replication, and they are different, even though they are symmetric and identical. they are, well, identical, they're complementary to each other, but the direction of replication fork breaks the symmetry. Same is probably true for some processes associated with DNA repair. So this means that if I run my statistical inference on the genome and I look at my intensity, I do not know about a specific direction of replication or direction of transcription, right? But if I assume that there is a process associated with transcription, as I will explain how this happens biologically in a little bit, then because we were not using direction of genes, we know nothing about the genome, right? And some genes would be oriented left and some genes would be oriented right. Intensity of the process that produces, say, A to G mutations in one strand and T to C mutations, say, in coding strand and non-coding strand, you would observe this intensity higher if the gene goes in this direction and low for genes going in the other direction. However, I would infer a different process, which would be truly complementary. It would go on the complementary strand and it would show the same kind of spectrum, but very complementary, right? So again, because my inference does not know about any biological mechanisms breaking symmetry between DNA strands, I would automatically learn two different processes, two different components, and two different spectra. But this spectra should be complementary to each other, right? So in this representation, we show a darker color and lighter color corresponding to complementary mutations. So here would be T to C is in dark green, and A to G would be in light green, and same here, for example, right? So C to A is in dark blue, and G to C would be in light blue, right? So this is what would happen. So this means that any process which is not noise, which is truly derived from data, can be either strand independent or strand dependent, right? If the process is strand independent, it should correlate with itself. If I would do the operation of complementarity, so if I would change the strand to reverse complement, the intensities should correlate. And I know two things, right? I know that this is not noise because this is the property that would only exist for a true process. And also, I know that this is a strand-independent mechanism. However, if the process is strand-dependent, then there will be no correlation, right? Because it depends on the strand, so there's no reason to expect that process would correlate with its self-complement. However, I know that I'm supposed to generate a pair of components, and I should find a component which would be complementary to my component. So this means that mutations in one component would be correlated with mutations in another component on the complementary strand. Okay. So what this allows me to do, this allows me two things. This allows me to know how many processes I'm supposed to find, and also this allows me to find out whether they are strand dependent or not strand dependent. So this is all about methodology. I'm moving to the data. And we used rare single nucleotide polymorphisms, we call them SNPs, from TopMed dataset. And the reason we didn't use family sequencing is because the data are too sparse on family sequencing to run the inference. The whole process was validated on the MAT data, which is much larger data set, and also on the novel mutations in trio sequencing data. Actually, NOMAD is much larger data set, only in some parts of the genome, not everywhere. So we used 192 mutational types. So again, every trinucleotide. So every mutation is characterized by 2 neighbors and type of mutations. And we looked at 10 KB non-overlapping windows. And to obtain all the components, we factorized this matrix product. And we only retain signatures that pass reflection test and are also supported by Bootstrap. Surprisingly enough, a lot of signatures, all strongest signatures pass this reflection test. So they are either self-complementary or complementary to the other strand with very high precision. So this is reflection metrics for all the inferred components, right? So they are either complementary to themselves, or there is another component to which they are another component with which they are correlated on the complementary strand. Okay, so this is all the statistical machinery and inference. So what about data? How are we going to understand what kind of biology produced these observations? And here we turn to epigenomics. We look at process intensities. So those processes that are generated by 2 components. So when we have strand-dependent processes, we can compute the intensity and we can compute the asymmetry. We can relate both intensities and asymmetry, and for strand-independent processes, of course, this is just intensity. And we compute correlations with gene bodies, with analysis of replication timing. So with places where DNA is being replicated early versus places where DNA is being replicated late. Acetylation marks, various epigenomics, methylation, position of line elements, recombinations, and so forth. And of course, direction of transcription and direction of replication. And we observe very strong correlations of individual processes with specific epigenomic measurements. And those are sufficient to generate hypotheses about biology. So at the end, we were able to explain or partially explain 7 out of 9 processes we discovered. And I will talk about some of the stories very briefly. I know we don't have a lot of time. And we explain some of them as driven by DNA damage, some of them as being driven by errors in DNA replications, and so forth, so on. So I will quickly mention a few stories. So this is one example. We don't know a lot about biology, but this is a strong correlation with timing of DNA replication. So this is process number 7. And this is its spectrum. And this process is extremely well correlated with replication timing. We don't know whether replication timing determines biology of this process or it's some other characteristic related to replication timing, but this is what we observe. Now, the most common process, which we call 1/2 because it has component 1 and component 2 and is strand dependent. So this process happens to be associated with transcription. So if you see there's 2 parts of the process, or component 1 and component 2, and this is direction of genes, again, the statistical machinery, the machinery underlying this inference was never informed by direction of genes or even location of genes. However, you see here clear symmetric pattern of intensity along the chromosome related to direction of genes. The reason we believe mutagenesis could be associated with transcription is the mechanism called transcription coupled repair. So as many of you might know, nucleotide excision repair is DNA repair mechanism. That repairs bulky damage. Bulky damage means that some DNA lesion that is not possible by replication fork and is not possible by transcription. There are two ways to recruit this downstream mechanism. One is global genome repair, and this is where a specific complex scans DNA, finding lesions. And another one is associated with transcription, right? So if RNA pol 2 transcribing the genes stops at the lesion, so it cannot pass the lesion, it recruits the downstream machinery of nucleotide excision repair that starts with helicase TF2H, unwinding DNA, and then followed by excision step and followed by resynthesis step. What is interesting here is that the same process is not only asymmetric with respect to transcription, but is asymmetric with respect to replication. And I think the reason here is the same. So the damage occurs symmetrically, but is repaired asymmetrically. So if you imagine replication fork, we have a leading strand with pol epsilon replicating DNA on the leading strand. And at the leading strand, it hits the lesion. It cannot pass any forward. So replication fork, there's no way to replicate DNA. So you have to stop replication, repair the lesion, and continue. However, on the lagging strand, replication is discontinuous. So if there is a lesion blocking progress of pol D, polymerase delta just starts another Kazaki fragment, bypassing the lesion, and this can be later fixed by translesion polymerase, which is very mutagenic, right? So again, we have symmetric occurrence of lesions, but asymmetric resolution with a repair system primarily driven to the leading strand. We confirm this mechanism looking at different cancers where we know the source of the lesion. So for example, UV type mutations generated in melanoma and smoking mutations generated in lung cancer. And we also observe this asymmetry. We also look directly at the analysis of XR sequencing data and damage sequencing data. And this comes from Sarkar lab, the Nobel Prize winner of Turkish origin, who some of you might know, who developed this amazing system quite recently by mapping the activity of nucleotide excision repair system along the genome and also sequencing the damage. So the idea is to generate crosslinks and identify positions of TF2H on DNA. And there is a similar assay to identify position of the lesion by, again, it's a sequencing-based assay that finds location of the lesions and find their genomic positions. So if we, when you look at this data, and these are fibroblasts that were hit by UV, you see what's happening, right? So this is, these are transcribed regions, and this is the activity of nucleotide excision repair system. And above, you see the amount of DNA damage, and this is function of time. So DNA damage specifically in places where transcription is high and the activity of nucleotide excision repair system is also elevated, is being cleared. So when we look at our data for this process 1-2, so there's two asymmetric components, we observe very strong correlation between results of the assay and results of our inference. So this means that, so we believe that this process is characterized by bulky DNA lesions that are not possible by either transcription or replication, and is strand asymmetric, and that's probably one of the leading sources of mutations in humans, and and it's not associated with misincorporation of nucleotides during replication. Okay, so the process which is probably associated with replication is the process that we call 3-4, and the reason, again, it's called 3-4 because it's strand asymmetric, and the reason we believe that this process is or might be associated with replication errors, because it correlates with direction of replication fork and pretty much nothing else. So it's fully explained by direction of replication fork along the genome. Another interesting process is, there are two processes associated with CpG dinucleotides. And CpG dinucleotides is a known mutagenic context, primarily driven by methylation of cytosines. As some of you might know, 80% of cytosines preceding guanines in the human genome in the CpG context are methylated. And deamination of cytosine transforms it to thymine, and this, at least a plausible explanation for this process, which we call component 10, and this is a process of CpG transitions. However, we also observe a lot of CpG transversions, and that was interesting. And when we look at this, we see that unlike CpG transitions in process 10, this process dips in CpG islands. This process spikes at CpG islands rather than dips, right? It's clear why process 10 dips in CpG islands, because methylation rates are lower. But at process 11, CpG transversions actually spikes at CpG islands. And we think what's happening is that this process is created by the mutagenic effect of active demethylation. And we have many other reasons to support this hypothesis. Okay, so what's happening here if we have methylated cytosine? We can have pure deamination, and this creates thymine, or if it's oxidated, and at the time of demethylation, there is a basic site created because it's being removed by the demethylation machinery. This frequently creates mutation to G or into A. And what this suggests active mutagenic effect of the demethylation machinery. And that, of course, affects very small fraction of the genome, but surprisingly important fraction, because it primarily impacts regulatory regions. And also, when we look at mutations, at early mosaic mutations, so mutations arising in early zygote, we see excess of this process. And that's probably related to developmental events like demethylation wave early in development. So this is what we believe is happening here. Okay, I should probably wrap up to leave some time for questions, but the last story is about clustered mutations. So our group and other groups, maybe 5-6 years ago, discovered that a number of mutations arise in clusters. About 2% of mutations arise in clusters in humans, so multiple events in the same chromosome within 50 kb. And what's interesting is that most of these clusters are inherited from mothers, not from fathers. And those are probably related to some process occurring in the non-dividing oocyte. We have some ideas what this process might be, but it's too early to present to the public because we're not quite sure yet. But what's important for this work is that when we look at intensity of our process, so a statistically inferred process, it has the same exact peaks with a large number of clustered mutations inherited from mothers. Again, the statistical inference machinery never knew parent of origin. So we clearly finding characteristic of a process that occurs in oocytes and is unrelated to cell division. And this process is associated with fragile genes and is highly asymmetric with respect to transcription in these genes. But what is also important that almost all effect of maternal age is explained by this process, right? So again, oocytes are arrested, cells are not dividing. However, this particular process increases in intensity between younger, not increases in intensity, increases its effect between young mothers and old mothers, right? So it has time dependency, and this time dependency doesn't scale with cell divisions. So we think that this is an active process acting in non-dividing cells. And it is associated with DNA damage, again, as shown by dependency on transcription. Okay, so in conclusions, so we have, I know this is a very technical presentation, but there are a few ideas I'd like to get across. One is that unsupervised statistical methods are now highly effective in genomics. We have plenty of data, and with this amount of data, we can use unsupervised statistical techniques to learn functional biology. So using this, we identified 9 processes that collectively explain genomic variation mutation rate, and I think we have a good handle on biology for 7 of them. Quick acknowledgements before we move to the questions. So I thank all of the lab. So these are members of current members of my lab. So I thank all of them, and specifically people involved in this work, both from the lab and from outside the lab. So the main 2 first authors are Vova and Ruslan. And it's primarily their work. And this is the lab over the years. And you see how we think about our projects. Well, we actually are looking at the seals here on Cape Cod. And this is our social events during pandemic, in winter outside, and now with restrictions being lifted and with the ability to gather outside. Thank you very much. So I don't know, I think we have a few minutes left for questions, if any.
Thank you, Shamil, so much for this very exciting and comprehensive talk. Now we can pass to questions. Please send us questions or ask just by raising hand. Questions, please. So we have one question in chat. Shamil, can you read this?
Right, so the question is, to what extent it is possible to classify disease-related mutations based on the structural and dynamic consequences in proteins? How would this contribute to personalized medicine? That's a great question. It's not directly related to this talk, but it's related to what we've been doing for years. And the problem is complicated. So let me elaborate on the question. So the question is the following, that there are multiple mutations, and some of them obviously happen in protein coding genes. So they impact proteins. And the question is whether we can predict which mutations are pathogenic or explain the effect of pathogenic mutations by looking at protein structure and dynamics. And this has been a relatively hard problem. There are many algorithms, as you might know, to predict pathogenic mutations. And most of the methods rely primarily on sequence data and sequence evolutionary conservation data and many other properties of sequence variation. Very few algorithms rely on structures. Structures commonly provide great insight, but the ability to predict is reduced. And this is quite surprising because a lot of mutations correspond to simple loss in stability. So you would predict delta delta G, so change of free energy due to mutation. And in the ideal world, that would lead you to the prediction. But it seems to be a hard problem. Now, I believe there is some new hope with AlphaFold and the ability of machine learning tools to move forward structural biology. So I think there is hope in this direction, but at least in my experience, very simple methods of using molecular dynamics or protein structural features don't move the needle to a large degree, right? So they are helpful, but not dramatically helpful. But again, I wouldn't discourage anybody from working in this area, because it might be that with the new methods, the progress is coming here. So I have.
We have another question from Ogün Adebalı.
Yeah, that's a great question. So what does explain the asymmetric repair between leading and lagging strands? So the question is whether stall polymerase inhibits repair. So that's a great question. So I spoke about two different mechanisms. So one is asymmetry associated with transcription, and that mechanism is easy to understand because polymerase 2 or RNA polymerase directly attracts the repair system to DNA. Now, why do we observe asymmetry in replication between leading and lagging strand during replication? So our model is the following, that repair is actually the same. However, what happens is that if there is a bulky damage on DNA on the leading strand, replication cannot go through. So the fork stalls. You have to resolve this, right? Because otherwise you cannot divide anymore. The cell would die. You can resolve this into, there are 3 pathways resolving this, but they are all error-free. But most likely you have to collapse the fork and repair, right? So the repair doesn't progress. In the lagging strand, because of Okazaki fragments and asymmetry and discontinuing synthesis, if polymerase delta hits the lesion, nothing happens. It immediately starts the next fragment. So there is no need to collapse the fork. The repair can still come and repair the lesion, but the kinetics of the process is such that replication proceeds, and there is no need, or there is no time devoted to attracting repair and repairing the lesion, right? So I don't know if this explains it.
Thank you. More question, please.
Right, so the question here is whether we can extend our methodology to other species, such as apes or more distant relatives, and get an idea about the evolution of these processes. That is an excellent question, and the postdoc in the lab who is hopefully goes on the faculty market, this is one of the idea that he will explore. We know that mutation rate evolves, we know that mutational mechanism evolves between species, and comparative genomics might be a very, very useful tool to explore this, right? So if we would find out what happened during evolution, what increases mutation rate, decreases mutation rate, changes parameters of the mutation rate, that would be extremely helpful. So the short answer is yes, I think it's a great idea. We might be looking into this at some point. We might benefit from samples of multiple individuals from different species, but I know Illumina, I think, is going to release, so there will be increased sequencing of multiple primates and other species.
Thank you. One more questions, please. Last questions, if possible. So, yes, great. Thank you. If no more questions, please, then Shamil, we can close the session. If you want, please give your concluding final remarks.
Okay.
It seems that no more questions, so thank you. People, thank you, Shamil. Okay.
Thank you very much. It was a pleasure to present here. Thank you.
Okay. Thank you. So bye. See you.